Om Namah Shivaya, Jai Gurudev. Today we will study about the central dogma in the molecular biology. So first of all, what do you mean by central dogma theory? So let's talk about it. So you all knew that our information carried out by the heredity, and the main factor of heredity is the DNA. So for structuration of the cell, the main composition is a protein. So however you think about how the protein is synthesized, so the answer is that protein is synthesized from the DNA. So DNA is the main factor for our heredity, and it's also play important role for the synthesis of the protein. And the technique by which the DNA synthesizes the protein is known as a central dogma. So let's talk about the steps of the central dogma. First of all, what happened? So DNA is synthesized from own molecule of the DNA. So DNA synthesized from the DNA. That's why it's known as a replication, replica. Not what do you mean by replica? The same structure, same function, and the same morphology and same anatomy of the DNA from the DNA. So parent DNA, which complete amount of the information that known as a template DNA, and this template DNA form new DNA. Here you can see that the first figure, DNA to DNA replication, then the synthesis of the RNA from DNA, that known as a transcription. Second time, the synthesis of the RNA from DNA, it's known as a transcription. And the last point is that the protein synthesis. The protein synthesis carried out from the RNA, that known as a translation. So the whole procedure of the replication, transcription and translation known as a central dogma theory. Now what is a gene is expressed in two steps. What do you mean by gene? So the protein synthesis. First of all, protein synthesis carried out from the DNA. DNA is transcribed to RNA, and that RNA translated into the protein. What is DNA replication procedure? Let's talk about the method of the DNA replication. So first about the history of the DNA. You all know that the structure of the DNA is a double helix shape. In 1950, Rosalind Franklin, by the technique of the X-ray photography of the DNA, the structure of the DNA knew, and then after the Watson and Crick, they described the whole DNA molecule. By the technique of the X-ray diffraction, the Franklin discovered the structure of the DNA. Now, what is the full form of the DNA? DNA means deoxyribonucleic acid. Deoxy, De means removing of oxygen, and whenever you write the spelling Di, it means two. So here you have to attention that deoxy means removing of the oxygen from the DNA second position. It's made up of the nucleotides. DNA double helix structures made up by the nucleotide. Now how polynucleotide form? By chain of the nucleotides, polynucleotide form, and these three molecules bind to each other and form the nucleotide molecule. So you can see in the figure, first is the phosphate group, second one is a sugar, which is deoxyribose sugar, and third one is a nitrogenous base like adenine, guanine, cytosine or thymine. If the structure of the RNA, then rather than the thymine, uracil base will become. So it's all about the central dogma and the replication. Thank you very much to all of you. Goodbye.